Welcome everyone to this seminar on European food law. My name is Mark Talon. I'm Managing Director of the food law consultancy Legal Foods. We specialise in all aspects of European food law, covering all 27 member states plus the UK. Today we're going to specifically focus on the novel status of cannabidiol or CBD. As it relates to its extraction from the Cannabis Sativa plant. The last 18 months have been quite an interesting time for industry, and it's fairly broadly agreed now that novel foods dossiers will need to be submitted to have CBD granted approval for sale in foods across Europe. To that end, I want to try and provide some guidance really here for industry when it comes to discussing these issues with trade associations or consultancies over what specific criteria needs to be covered as regards a successful novel foods dossier application. Now, these can be quite complex and definitely expensive. There's been a lot of misinformation in our view over what the requirements are for such dossiers. We hope this provides some clarity on those issues, and if you've got any questions, feel free to reach out to us. The presentation is about 60 minutes in length, so grab a coffee, and I hope you enjoy the content. Thank you for watching. Welcome everyone. This is our seminar on novel foods, the facts and fees of CBD submissions. So a very quick overview of Legal Foods. We specialise in European food law, specifically 27 member states plus the UK. There's a selection of different areas you can see there on the slide that we work on. We've been doing this for quite a few years. Here's a few of our clients. I don't want to spend too much time on telling you about the company and exactly what we do. You can find out more about us on legalfoods.com. But to say that we have been involved in novel foods work for many years, advised a number of different clients over not only individual ingredients, and their application in different foods on different markets, but as well as the dossiers themselves. A little bit about myself. So I'm Managing Director of Legal Foods, and I have been for the past 12 years. My background is in nutritional biochemistry, and studied that at a number of different institutions. More recently, my specialist areas of academic training have been around food law, with masters in food law and in European law, and I hold a number of different affiliations through different institutions, specifically on the food side of the industry. So today's talk, there's quite a few different topics that we need to try and get through. But in general, I want to try and direct this at ultimately sort of non-scientists. So we cover sort of a broad range of different food companies and skill base within each company. So from those that are to do with sort of much more pure legal issues, to brand managers, and everywhere in between within food companies. So the first two topics that we are going to cover are novel foods regulation. So just a very quick general overview. Why we're actually looking at this issue. What is toxicology, for those who don't know that, and how that relates to this specific seminar covering CBD. And then we basically want to get into sort of the meat and bones of what's required as regards to novel foods dossier. And I'm trying to squash this down really to sort of 7 key areas that I think are definitely the parts of a dossier that need to be understood. And this should help provide some sort of general guidance over these issues. Now, I will say from sort of the outset, there's lots of guidance out there, and I would advise any sort of food company to look at those guidance documents and to see what's exactly what is involved, and specifically the requirements for these different areas. One thing that is needed is sort of an understanding of what's happened historically over novel foods and the way these dossiers have been assessed. So you have sort of the general guidance documents, and then I would sort of advise to then look at specific novel foods dossiers to see exactly what's acceptable by EFSA, so European Food Safety Authority, or the FSA in the UK. And then to sort of wrap up, I want to look at sort of some practical side of things. So what are the typical costs involved with the construction of these dossiers? So we do see some broad-ranging costs associated with their construction, and some of these costs are definitely misleading. So we'll sort of try and give a more accurate overview of what costs would be involved, depending on the type of dossier and the areas that you want to look at as part of that dossier, what the options are for business, so whether that's sort of an individual submission or consortium, or do nothing maybe, and then down to sort of choosing experts really. So who would you look at sort of within the industry, and what you should be asking for when you choose someone to sort of construct these dossiers for you. So the Novel Foods Regulation really has sort of a dual purpose at a basic level. So like most regulations or legislation at a European level, it's to maintain an effective functioning of the internal market. So harmonization basically across the different member states, and to make sure that ultimately sort of most companies work on a level playing field, and that enables sort of the free movement of foods within or across member states. And the second area, which is specific to what we're going to look at today, is to provide a high level of protection of human health. So in basic terms, that means the safety of any food products that are placed on the market, and how we can provide protection for that under the Novel Foods Regulation, and its framework for assessment of food products. So why this presentation today, and why on cannabidiol? Well, I think we can probably all agree at this point that cannabidiol, or CBD, as an extract or an isolate, is considered, in the eyes of the regulator, novel. It really doesn't matter what I believe, working in the area of regulatory affairs or food law. Ultimately, the most important bit is what the regulator has decided. And it's clear that at a European level and a member state level, these products are being classified as novel. And that means they won't be allowed to be placed on the market without prior authorization. And it comes down to one simple thing, that they don't believe that prior to the 15th of May 1997, these extracts or isolates were on the market. And therefore, to get them approved, we need to construct a novel foods dossier to show X dose is safe in X food application. And that's what we're going to look into. So toxicology is the sort of underlying aspects of any novel foods dossier. So what we're trying to look at here is the study of adverse effects, or in what I like is a sort of a better view of is the absence of an adverse effect when we provide a given chemical. So we often forget that foods are made up of multiple chemicals, and what effect those chemicals have on you and me when we take them in different dosages and different food forms. Now we must remember that the dose ultimately makes the poison. So we often hear about X product, we need to deliver a certain amount of an ingredient to deliver an effect, and often there's that relationship between an increase in the amount of a dose of a product and its efficacy. But likewise, as the level of a compound or ingredient or chemical is increased, often too is its level of toxicity. So what we're trying to do is, as part of this toxicological work, is to find out what level of that ingredient, as an isolated ingredient or in a specific food application. So if that ingredient was put into a beverage, into a bar, into a supplement, what level is safe, and what is the sort of recommended daily usages for that product. So often the starting point for any ingredient that's going to be used in a novel foods dossier is something called characterization. So ultimately, is that ingredient what we say that it is? So to prove that, we need to identify the ingredient, and there's multiple different methods of doing that. So we can see here basically on the right, this is called a chromatogram, and there's a series of different chemicals there, 10 cannabinoids that we find in the cannabis plant, and we can see the one that's highlighted in green is CBD, and that's run against a series of standards. We can sort of pull that out and we can quantify that in terms of how much might be in the product, as well as its ratio to other substances within that product as well. And what we're trying to do here is to demonstrate, and this is sort of the key issue of any novel foods dossier, is that the chemical that we identify as what we want EFSA or the FSA to look at in the UK is the one that we're going to actually use in our toxicology studies. So we want consistency of data through any type of assessment. So there's no point saying, oh, well, we're going to look at a CBD isolate that's 98-99% pure, but the one that we actually run in the study is only sort of 90% pure, and it's got lots of other different cannabinoids in there. That causes huge issues and ultimately rejection of studies. So we need to make sure that we identify what the actual chemical is that we're going to look at, and make sure that chemical is going to be in our final study. We should, as part of that, we should use the same production process. So our manufacturing, we need to look at the source materials, what are the residues that we get as part of that manufacturing process, and contaminants, and all that information needs to sort of to be put together as part of this characterization work. The next sort of important area is what we call stability. Now, stability ultimately in sort of lay language is our ingredient, either by itself or in a finished product. How long does it last in that state under certain storage conditions? Now, there's different tests that we can do to actually prove the stability or lack of stability of that ingredient in its form that we're going to submit for a novel foods approval. And we should consider lots of sort of different issues here. So you can see within this definition that they're going to look at, you need to look at things such as, so this is not sort of catch-all definition, but things like temperature, humidity, light, all of these things might have an effect on how stable that ingredient is. So how much is left of that specific compound at the end of its shelf life. and that's tied in really to sort of the recommended storage conditions or the best before date or use by date that you're going to put on that product. And that again will be sort of included in your novel foods dossier. So what do EFSA say about sort of the stability work that needs to be carried out as part of a dossier? Well, there's not a huge amount relative to other aspects of a novel foods dossier construction. They're giving guidance, but it should, for example, stability work should be carried out under normal conditions. So that includes sort of what type of packaging the final product's going to be in, how it's going to be sort of sold. So will it be in a refrigerator? Will it be on shelf? Is the packaging opaque? So could sort of light have an effect on the packaging? Is it sealed? Can air get into the product? So all of those type of issues need to be considered as regards the stability work that you carry out. Preferably, this should cover sort of 5 representative batches of the novel food that should have been independently produced from raw materials, and it should cover at least sort of the shelf life up to the end of the shelf life of the product. So as part of the submission, you'll say, well, it might have a 12-month shelf life or an 18 or 24-month shelf life, and as part of that, that's the stability work. So you would sort of take samples of the product at different time points, and you would assess how much, for example, cannabidiol is left in the product. But you might also look at other substances. So what can CBD convert to, and what other ingredients might be present in that product. And specifically the ones that are going to go into the toxicology study. Other aspects that you should think about is if you're going to put sort of put the ingredient into other foods, could that also sort of influence the stability of the ingredient that you're applying for a novel foods application for? So for example, there's no point just assessing a CBD isolate if it's then going to be put into a product that might alter the pH or the level of humidity of that CBD is going to be exposed to. The same again, it could be sort of also its exposure to light. So we know from some studies, for example, if CBD is packed into a bar, it's only sort of the external part of the bar that's exposed to light. So that might have a different type of effect to a product that's actually just put into a glass with some liquid, where more light may get through to all the different, a much more amount of CBD than in a bar product. So this is a sort of relatively simple summary of what we think, at least some of the aspects to consider. So you can carry out accelerated stability work. This is where you would basically use a higher temperature to accelerate the conditions that the CBD would be exposed to. That would be typically done for sort of 6 months, but we would really sort of advise that real-time stability work is done, and this is sort of an issue to anyone who's sort of late to this process, unless you've actually carried this out already. Real-time, depending on the shelf life that you're going to advise for in the final product, it can be anywhere from sort of 6 to 36 months or even longer. And again, it should be sort of in the same conditions that the product's actually going to be held for sale. So if it's going to be in a liquid that's in a chiller or a refrigerated food format, those things should be considered. Again, 5 batches of the novel food, using sort of the same manufacturing process that the final product will use, and you need to give sort of details on manufacturing. The storage packaging, So this should mimic the same sort of closure system you've got in the final product, where it's going to be distributed. The evaluation frequency, so basically what time points are you going to take samples of the product for chemical analysis to see exactly what's happening to that material. So if you still got if you start off with 98% CBD, what happens at 3, 6, 12 months or even longer to that level of CBD? Does it convert into anything else? So those type of things would be looked at over those different time points. What to test? Again, these are sort of a superficial review of these issues because it really depends on the product, the type of extract, and ultimately things as simple as sort of the dose of the product and storage conditions can all impact what you would actually test for. But definitely should include sort of microbial, physical, chemical, and biological characteristics. And then we need to sort of tie that into safe limits, because remember, the whole dossier really is about, is the actual final product safe for human consumption? We can actually carry out some stress testing. So we might want to look, for example, at how does CBD degrade? So different pHs, different temperatures, different humidities. If for example, you might only want to market the product in sort of temperate conditions. What about if you put it in a very hot condition or a very cold condition? So all of these sort of issues are relevant and up for consideration. Ideally, we would suggest that you actually look at sort of at least some testing in the finished product. So there's no point sort of taking a very small sample in the batches and saying, oh, well, that's relative to the actual final manufacturing production. So if you're producing hundreds of kilos, that could result in sort of a very different mixture of compound versus only sort of less than a kilo. So there's a few different ways to look at that and to include that in the stability testing. So again, just the things to sort of think about is the light conditions, temperature. Is the product open to the air, especially on how to use the product? Do you need to consume within a certain period of time? What about acidity for different food uses? What's the humidity, or is it going to be in sort of a fluid? How might that impact microbial breakdown? What is the level of extraction? So we know that there's some data there already on a higher amount of the extract versus a lower amount of the extract can affect stability. Packaging size. So small flasks and vials have a small surface to volume ratio, so they have more light exposure, and again that would affect stability. So all of these things are sort of tied into that stability work and definitely open for consideration when you're thinking about a study. So when you actually discuss these type of issues with a consultancy or a trade association, they should be taking all these type of issues into consideration about your specific product. And that's what will ultimately make it difficult when it comes to consortium submissions, because every company might use a different type of packaging. So all of that would need to be considered for any type of submission. So just before we sort of jump into the specific toxicology studies, unfortunately, animal studies are still a requirement as part of novel foods work. What animals are used in the study is sort of another issue, but there's definitely sort of a push now towards reducing, the pain and suffering of animals as part of these studies, and that can be done from a study design point of view in terms of what dosages you're giving the animals and how you treat them, and there's some specific guidelines on that. And whichever lab you use to sort of carry out your studies, there should be foreign sort of well-established guidelines for the humane treatment of animals during these type of trials. So we're going to look at sort of now three main areas of novel foods dossiers, and these are dose range finding studies or citing studies. We're going to look at what's known as ADME, or absorption, distribution, metabolism, and excretion. So what happens to CBD when you consume that? Where does it go in the body and how is it excreted? And then sort of toxicodynamics, or the OECD guidelines, which is the one that's principally used in most novel foods dossiers and including additive dossiers to show whether something's toxic or non-toxic, or should I say what dose is non-toxic, is the 4-8 study design. So most of the different aspects of the novel foods dossier as regards actual study work itself are based on a sort of tiered program design. So at each tier, we're building on sort of information based on assessments each time. So we only move up to sort of the next level of assessment based on the information found in the one that we've just done. As part of this, we've got this whole 3Rs side of things, which is about the ethical treatment of animals. So we're trying to sort of reduce the dose that they're exposed to, and refine these study designs to try and sort of not have an animal experience undue levels of pain. And there is sort of like a significant move now for these type of designs for the better treatment of animals. We're not at the level yet where we can actually just do these studies only using sort of mathematical modelling. Hopefully one day we will be, but for now we will need to sort of use animals for this type of testing. So a dose range finding study is ultimately to try and find in a very acute way, so from a single dose to repeat dose over sort of a 14-day period, is to try and come up with a dose that we're going to use in the larger study, so the either a 28-day study or the 90-day studies that we're going to move on to. And this is also a good time that we can actually carry out our ADME work to see how it's sort of metabolized within the body and how it's excreted. So here's a sort of general overview of what ADME is. So it's absorption, distribution, metabolism, and excretion. So the distribution is related to sort of where the CBD goes within the body. Now this can be sort of stored in body fat, which is sort of really important area at the moment, and we've got some limited data there, but because it's lipophilic, like most cannabinoids, it does get stored in lipid membranes, including adipose tissue, and that could mean that it accumulates in the body, and then that could ultimately have sort of longer term implications if it's going to be in the body beyond sort of a 90-day toxicology study. There's also additional studies now that are starting to look at what happened to the accumulated CBD or other cannabinoids and other compounds from the cannabis plant, like THC. What happens, for example, under fasting conditions, during exercise, under stress conditions. It seems like these different physical states can affect the release of CBD from the body. Metabolism is another area that would be looked at as part of ADME work. So, what's its bioavailability, what happens to it, how does it actually pass through the body and get to sites of action. The absorption, so how does it actually get out of the digestive system, through the intestine, through the gut, and then into the circulation. And we can look at obviously what those release rates are by appearance in the blood. And then we can do obviously some pharmacokinetic work to look at sort of where the maximum level occurs, what the area under the curve is. And finally, excretion. So how does it get out of the body, and what is sort of the time frame for that? And there's a few different ways that we can look at excretion. So what options have we got for ADME studies? Well, as I was trying to sort of say earlier, we can do some mathematical modelling or in silico work. It has sort of some limited application, I think, to sort of CBD at this point, with it being a relatively new compound, and we definitely don't know enough about how it's sort of stored in different compartments of the body to actually fit a proper mathematical model to that. So the take home is ultimately we're going to need to do some studies in humans or in animals, and then potentially some additional cell culture work, depending on what we really need to see and what we get from the early work from the human studies. Again, this is sort of a tiered approach. We have some guidelines, so OECD 417 would be useful. And there's often, in a lot of the work from sort of a novel foods point of view, you have sort of theoretical versus experimental approaches to how we actually model how CBD or any other sort of compound is metabolized within the body. So we can look at published data, but in my view, we will still need some specific studies on your compound, with the vehicle that's used for absorption of that product. Those are definitely things that we're going to need to look at in a novel food study. So the dose range finding study design, there's a few different testing options. So in vivo or in an animal or in a human. Most of these are sort of rodent studies initially. So we can do sort of a fixed dose trial, and there's some additional detail on this slide. There's acute toxic class. There's up and down methods, and there's some others, but you can see there's some references there for those that want to look into this in a little bit more detail, and some guidance there on the OECD guidelines. But ultimately what we're trying to do is come up with a good estimate of a dose that or a selection of dosages that are going to be sort of a low, medium and high, and that's what we sort of use in our final 4-8 toxicology study, and from that's where we'll come up with a dose that's going to be safe for use in the final product, and that's what we're going to put forward to EFSA or the FSA. So this is the sort of main meat and potatoes of the study. So sub-acute toxicity studies is the heading that it sort of fits under, and the OECD guidance 4.8 is the sort of bones or skeleton of these type of study designs. Now, what I say from sort of the outset is people reference to say, oh, well, we're doing our 4.8 study. Well, what does that mean? Well, the 4.8 is a basic design. It's not a comprehensive study design saying what you need for your specific product. It gives you sort of the base features of what a toxicology study would look like. So how many rats, maybe what conditions they're kept in, how you would sort of feed them, what things you would look at that relate to toxicology, so what markers we might look at. But these again should be sort of tailored to your ingredient, and also to what issues have already been raised in the published literature
The main thing that separates them from sort of a dose range finding study or citing studies is that these are repeated dosages. So, it could be multiple times a day for X number of weeks, or every day, for again, weeks at a time. And then what we're looking for then is sort of how that might relate to food use. So, ultimately, if your prescription is to take CBD once a day, then obviously the 4-8 study is much more valid, much more related to that type of intake. Very few people are going to say, okay, then take CBD for one single use and then that's it. So the 4-8 study is sort of the key for getting any sort of novel foods approval. And what we're trying to get from this is a longer term insight into the cumulative effects of CBD, how it's stored in the body, and one important area is its reversibility. So as I spoke about earlier, that CBD seems to build up in lipids or fats of the body. So we want a period after the 90 days, the 90-day study, that follows what happens to the CBD that may still be in the body. So we might look at sort of changes in blood plasma, blood levels of CBD, in urinary excretion, for at least sort of 4 weeks following cessation of the intake of CBD in the rodent study. So here we can sort of see what the studies really are all about. So we can see sort of a dose curve there. We can see 3 green dots. Now these represent different dosages of CBD. So milligrams of CBD per kilogram body weight. And then up on the y-axis, we can see some markers there that relate to potential toxicity or safety. So ALT or AST, these are liver enzymes. And we can see as the dose increases, so does ultimately sort of the levels of these liver enzymes. And what we're trying to find out is a point of departure from what we would consider as a safe level. So there'll be a point where ALT it reaches a level that's considered as unsafe, and moves away from what would be sort of a normal physiological range, and that might be then considered as the sort of point of departure, or sometimes you might see it in the literature as reference point. And there's a number of different ways of trying to assess where that point will occur. So we have sort of the lowest observable adverse events level. It is probably one that people know about. Then there's no observable adverse events level. That's probably the one that historically everyone's used in the past. And then over the past few years, we've seen sort of the appearance of benchmark modelling, and ultimately it's, in basic terms, it's a way of sort of fitting a curve through the different measures that we've got to try and get a more accurate level, to try and get a more accurate marker of where this point of departure would be using mathematical modelling. So here's sort of a general overview of sub-acute toxicity studies using the 4-8 design. So we can have sort of different durations. So it could be 28 days, 90 days, or over that time frame. We have different types of tests depending on what we're going to look at for the toxicity. So it could be sort of OECD 4.7, 4.8, 4.5.2. And we can see there on the right hand side, depending on what the study design is, we have different type of group structures, different genders, and then obviously the total number of rats that we use these studies can be massively different. So from, there's a tenfold difference there from using sort of a 4.7 design to a 4.5.2 design. Most of the novel foods assessments and ultimately the novel foods approvals use the 4-8 design, so a 90-day study, and then obviously following that you would have sort of a recovery period of a month. So you at least need to sort of build in 4 months worth of work just for the actual studies itself. That doesn't include obviously the time to assess, sign off on what protocols are used within that study design, and then analysis of any data as well. That comes out the back end of that, and any obviously repeated work that might need to be carried out. So it can be sort of very time consuming to do these studies. So as we briefly covered out earlier, the preferred design is the OECD 4-8, and obviously it has limitations, and there's definitely sort of other areas that you might want to look at. So what we do see in that study, there's no assessment of the ability of rodents to reproduce. There's no real sort of assessment of developmental effects. So yes, to sort of look at sort of external observations of reproductive organs, but they don't look at what happens, in generations. So there's other aspects definitely to sort of look at within that study design, and again, that's where your sort of consultancy or trade association should be feeding in to say, well, this is what we need to look at. These are the specific markers or the specific study design that we need to assess to make sure those are covered. And again, that'll relate to the dose for your product, the type of extract, the final product. All of these things need to sort of feed into that type of assessment of what needs to be added in. So what is an adverse effect? So we need to weigh up the difference between a statistically significant effect. So there might be some effect there based off the stats, but it has no biological significance. So then you can sort of come up with an argument if that was the case, to be able to say to EFSA or the FSA, look, it's statistically significant, but there's hardly any change of biological significance. And in that regards, that's where your sort of science comes in, to understand what are the strengths and limitations of different study designs. So for example, the genotoxicological studies, you have sort of a 471, a 487, 489 OECD design, and you should understand really what the difference is between those different studies, and what they're actually trying to assess, what the limitations may be. We need to make sure that we include a recovery group. So we add that again to this standard test, this 4-8 design. We look at the reversibility. So, for example, you might get some acute liver issues. So there might be some slight elevations in those markers that we looked at earlier. So these sort of ALT, AST levels. But over the space of maybe 28 days, those levels have dropped back down to normal, and that's why you include this recovery group. Other testing, as I sort of briefly covered there, so genotoxicity. So there's multiple different tests. Do we do all the different OECD tests, or do we select one, or do we select two? So again, this is where sort of the external expertise should come into this. If you're going to look at sort of additional studies outside the 4-8 design, so if you're going to look at reproduction, what type of toxicity tests would you look at? Analysis of the results. So you need to sort of consider what type of approach you're going to do for your analysis. Again, you may want to consider then obviously how many groups you've got in your study. So some suggest, for example, you might want 6 groups at different dosages to do proper benchmark design type analysis. Others are happy with 3. So you need to know the strengths and weaknesses of how that analysis is carried out. And again, obviously, all of these things are going to be tied together with things like cost. So, it sounds beautiful to have a nice data set that might be 6 or 7 different dosage groups, but that may fiscally just not be possible for many companies. So the standard is sort of 3 dosage points, and then we have to think about how we're going to analyze that data. Uncertainty factors. So uncertainty factors are things like when we actually come out with a value in terms of milligrams per kilogram of CBD that's toxic in the rodent, you then have to correct that to try and extrapolate those values to what might be toxic in a human. And these are called uncertainty factors or point of departure modification corrections. So typically you will have sort of an interspecies level. So that's sort of a correction from rat to human, and then you'll have sort of interspecies, so between humans, and then you can have lots of other ones depending on how good your data is and the type of test you've done. So you can have correction factors to predict from subchronic to chronic use, and then things like, for example, if you've only got sort of a low final marker versus no, again, that can bring about additional correction values. And the take home ultimately for these sort of uncertainty factor corrections is that every time there's one applied, you end up with a lower and lower dose in your final product, and in terms of what's considered as safe. And this is another reason why you really need to think about what the study design is, how many sort of different dosages that you need to be assessing, and what the actual 4-weight study design is, and even sort of further back in the sort of citing studies or the dose range finding studies, you can sort of start looking at the literature and trying to make some decisions about, well, okay, we've seen this at this dose, maybe we want to use X dose in our sort of dose range finding studies. And again, this comes down to sort of the expertise that you use in construction of the dossier. Another part of the novel foods dossier, so a lot of people just think, oh, I only need to do the toxicology study. I only need to do a characterization study or stability study on the ingredient itself. But these are sort of, the dossier is massive. I mean, it's just, those are sort of, yes, important parts of the study, but we also need to look at sort of the published literature. So what everyone else found already as regards to safety or non-safety of cannabidiol or cannabis extracts. And as part of that, you need to carry out what's known as a systematic or comprehensive review of the published data. Now, member states have done that in part. So COT studies, the WHO study, the Australian study, the TG study. So there's some that have been done with certain limitations, but you need to think about what the limitations are going to be needed for this submission based on your specific ingredient. So again, there's guidance there on what's required to do the literature review. You need to think about what are the relative, or should I say, the relevant safety outcomes to look at those studies, what your inclusion and exclusion criteria are going to be for that search. It may only consist of assessing cannabidiol, but it really depends again. So what's in your final product? So has it got other cannabinoids in there? What happens from the stability work? So when you do the stability stuff, does THC appear in the product? So if you've got, for example, exposure to light, the light may convert the cannabidiol to THC, or if it was more acidic. So there's sort of lots of things you need to think about as regards what might need to to go into this sort of comprehensive literature review. And again, all of these things feed in together to try and come up with a general consensus about what dose is safe in the final product. Another area is food juices. So it's great if you've only got a capsule and you've got sort of your CBD isolate in there. It makes it quite simple. But ultimately, if you've got sort of multiple food uses, so if it's going to go into a bar, if it's going to be in a beverage, then we need to consider what is a typical sort of consumption pattern for those type of products, and what that might do as regards a population intake. So EFSA has a few different sort of tools that can be used in that regard. So Food X2 is one of those. We also need to consider if it's nutritionally disadvantageous. So again, consider what the actual final product is that the CBD is going to be used in. And then we need to put that in the final dossier as well. So I guess the important section here is costs. So we see sort of all sorts of costs bandied about online in other presentations that we've seen from other consultancies, especially when we see presentations from labs or trade associations. It's, in our view, a lot of this has been quite misleading about what the costs are of dossier construction. In our view, ultimately, you need to think about this in terms of the whole project. A submission, a novel food submission based on the published data, in our view, is highly unlikely to be approved. Highly unlikely. My question would be to those that are suggesting, we'll put a dossier together based on the published data, and all you need then is to do some stability work, is show me some novel food studies that have used that approach and where we've seen approval. In general, it's that type of study has only ever worked when we used to have a method called substantial equivalence. So someone had already done the hard work in terms of they'd done sort of the 4-weight studies, and then someone would piggyback on that data to say, we want to now use that in a bar, or we want to use it in a beverage. So it was just a different food use. So I think it's highly unlikely that a literature-only submission will be approved. Fantastic if it does, and well done whoever puts that dossier together, but we wouldn't be doing that type of dossier for our clients because we simply think that it's just going to be rejected. But I'm sure there'll be someone out there that's willing to take some cash and tell you whatever you need to believe to think that you don't need to do an expensive sort of toxicology study. So we think based on the construction of sort of a literature only assessment and submission, again it comes back to sort of what the compound is, but let's say it's a CBD isolate, you're probably looking at somewhere between 40 to 60k to do that type of study. Then if we look, then this basic versus optimal, so what that means is sort of a basic 4-weight design with some stability work, a little bit of characterization. You could be anywhere sort of between circa 300,000 UK pounds. And then obviously if as the study design is amended, so that means if you're having a recovery period plus additional markers on developmental issues, reproduction, other maybe some novel markers that you might want to look at as part of that study design where we think from the published literature there might be some issues, you can be up towards the 500k mark very quickly, and that's sort of what we would suggest that you need to be considering before you sort of start moving down that route. Other issues are ultimately who's going to advise you on these type of things. So you might have a contract research organisation that carries out the actual study, or they may sort of outsource to a university but provide guidance on that. You might use a consultancy like ourselves. And again, sort of there'll be an additional cost involved for those type of companies that are going to advise on top of sort of dossier construction costs. So you need to consider that. So what are the practical options that are open there to industry? Well, we think there's sort of really only three options, and there's pros and cons to different types. So you as an individual company can do a submission, a novel food submission, and as we've sort of spoke about earlier, can be extremely expensive. But you would own the IP, so you would own the rights to that submission on that specific CBD product, that ingredient, and only you would be able to sell that product upon approval. No one else would be able to actually sell CBD using any other manufacturing process, only if they actually purchased it from you or from whatever supply you've actually used as part of that dossier submission, which means ultimately you could actually sell business to business use of your ingredient, or you actually then control part of the market for a certain amount of time before anyone else then can actually get a dossier approved. You have legal certainty, which means that once it's signed off at an EU level rather than just a UK level, that you can sell that product across all different member states under the conditions that have been approved as part of that novel foods assessment. Of course, there's always going to be a risk of submission in the final dose, and that's for sort of discussions early on before you start the study. So you could end up with so if you're selling a product that's got a really high dose of CBD in there, or that's ultimately you're going to be your model over the long term, what is your sort of view if you only ended up with a product that's only got sort of 5 milligrams, 20 milligrams, 40 milligrams? What is the dose that you're only going to accept in that final outcome of novel foods dossier? And you can definitely do some sort of early risk assessment work to say, well, look, at this point we think this is maybe a safe level that might get through, but ultimately the sort of dose range finding or citing studies will help provide some additional guidance on that before you jump into a fully fledged 408 design. So you might want some break clauses in there to make those considerations. The widely touted consortium approach, so this is sort of pushed by trade associations to a fair degree. We definitely sort of see some benefits in it, but there's also a lot of cons in there, and on at a business level, we can see sort of some real headaches coming up. So the pros, shared costs. So again, we said that sort of the upper level might be 500 to 600,000 UK pounds for the full dossier, including its construction. Having that split between sort of multiple business, it's sort of a no-brainer. That's definitely sort of a benefit. You might get sort of pooled knowledge or sort of insights from the different companies about how they think the sort of dossier should be put together or what should be included. There's potential, obviously, to tap into even more funds because the amount shared. So you could have sort of wider studies that might look at additional markers. And ultimately, obviously, media attention because all of the companies work together. You can sort of get more focus, more attention on the actual submission itself and what the benefits might be, and that might have its own value for a company, especially if you're a publicly listed company that's selling CBD. The cons for that side of the approach, I probably could have filled 5 sides with the cons, but I'm going to stick to sort of the main points. So who owns the data basically is going to be a real issue. So if you're all involved in the submission, there's going to be there's going to have to be a pre-agreed contract about who owns the submission and how that's going to be shared. So that means contract lawyers are going to need to be involved to try and broker some type of deal before the actual dossier is submitted. That may raise some contractual issues over intellectual property that are open for discussion. Pooled knowledge, although that's also a pro, it could be a con where different companies are fighting against each other about what should be in the dossier and what shouldn't be in the dossier, what the specifications are of the product, what the final food uses are, and ultimately all of those type of issues could slow down the decision making process. And as we know, at least for the UK market, you've got to have a validated dossier for the end of March. And if that's not occurred, products will begin being removed from the market. And obviously, all the other different member states, it's already considered as novel and not lawfully for sale, although it is tolerated in some markets. It could cause sort of more issues. And the final one, ultimately, you could do nothing. So don't do a submission. Continue as many sales as you can through to March 2021. And hope that someone gets an approval, that's a supplier, that can meet your demands, and you can sort of broker a good deal for someone who has invested in that process at that point. The risks of that obviously are if you can't broker a deal or no one gets an approval, then the product could be removed from the market, and that is the end of your business. You could be in a situation where you've got no other choice but to try and sell it as a novel ingredient. And again, that could then open you up to other enforcement action, including stop notices, fines, and again, obviously, potentially other studies might come out between now and then that show the dose in your product is unsafe. So then you could be open to sort of unlimited fines, criminal prosecution. There's sort of multiple potential issues there if you do nothing, but still want to continue selling your product when you know that it's novel. And obviously that can feed over into the media side of things. So if someone picks up that you've been selling a novel ingredient, that can damage business. So we're nearly there, nearly at the final section of this seminar, and well done for sticking with it. I know it's quite a dry area, but hopefully there's some useful information in there. So one of the big questions that I have with my peers in this area is how can sort of industry or food businesses choose what people are now calling themselves as experts in novel foods, and it's definitely an area of caution. We see a lot of companies that pop up that have maybe worked on medical devices or in the pharma industry, or biocidals or cosmetics, and now all of a sudden they're experts in food law. So I would sort of give some general advice as regards how to sort of narrow down who you might want to partner with in getting a novel foods approval. So as a food law expert, have they done any qualifications in law? Have they got any academic training in law? What makes them an expert in food law? Is it because they just say they're an expert in food law, or have they got any qualifications? So that doesn't mean sort of a 6-week online certificate. Have they got a degree, a master's degree, a doctorate, and that applies just as much as being a scientist. And again, obviously, there's the different levels within that. So, you could have qualifications in law, but not in food law, or European food law, and the same as being a scientist. So there's no point being a scientist, and you've never studied food. So you need to sort of assess someone that specifically looked at the type of methods So things like HPLC, for example, which is one way of assessing compounds. What type of expertise have they got in biochemistry or organic chemistry? Or have they got a team around them that they use as part of this work? And the same sort of legal qualifications. So just because you're a solicitor, for example, doesn't mean you're an expert in food law. There's some fantastic solicitors out there. So Hilary Ross, for example, from DWF, or Brian Kelly from Covington. These are people who've been practicing food law for well over a decade, and are experienced in this specific aspect of law. So it's pointless someone coming in who are reading some legal practice notes and thinking, okay, now I'm an expert in novel foods. You need to actually look at what they've specialised in, how long they've specialised in it. Is definitely sort of is key questions to ask. Practical application. So academic knowledge is definitely sort of one part of that criteria for choosing a partner. But how long have they been working in food law, and what is the reputation? So if people have sort of been working in novel foods, and specifically food law, have they got a track record within that category? What's a reputation like? What are other business saying about them? And you can, feel free to sort of ask those questions about the experts that you use. Don't just choose at face matter that someone says, oh, we're an expert now in food law, when they've got no history of specialisation in this area. So to wrap up and conclude, things to consider about actually going forwards with a novel food submission. What is your return on investment? So, ultimately it comes down to is it really worth spending potentially £500,000 on a novel foods dossier as an individual for that submission? Can you actually make the money back on that type of investment? And are you willing to sort of take some risks? It's quite a difficult one. And this is, I guess, why a lot of people are drawn towards a consortium approach, or what we think is sort of the misleading side of, a submission based on the published data. And I think what you might see is people do submissions on the published data, go away, do additional work, a real sort of toxicology studies, and then resubmit and ultimately sort of add to that type of submission. But we'll see. Who owns the data is definitely a key one, because you get some benefits if you submit individually. That can give you sort of a key market advantage in terms of making money from a novel food submission if you want to sell that product to other businesses, or if you want to be sort of the sole owner for that novel foods submission. But you've got to be willing to sort of enforce it, or actually push competent authorities to ensure that if you've invested in a novel food submission and no one else gets an approval, because you invested the time and the resources to own that submission, that other people aren't still selling novel foods, and that may again have additional costs involved to make sure those other companies are not allowed to operate within the market with novel CBD products. What food uses? So from the outset, consider what applications you want the products the CBD to actually go into. Now it might be a case that you, for financial reasons, you maybe only want to do stability work and characterization on only a number of food products, and then extend that maybe further down the line. Or it may be, your main focus is, it's only going to be in CBD in a beverage, and you tailor that down right from the beginning, and that's where your investment will go into. intellectual property. So this is sort of quite an interesting area. So if you're using data in a submission, you're going to have to consider who's funded that study. Are you going to be able to get hold of enough information from a 4.8 study that's published in a peer-reviewed journal to make sure the ingredient, the manufacturing process, the stability in that study is directly relevant to your CBD ingredient, to your final food product? These are definitely sort of all issues of interest. Is there a potential for infringement of other people's intellectual property when you do a published data only submission? What about using information from patents that may have been paid for by pharma companies? Is your actual CBD ingredient itself infringing a current patent? So is the dose or the composition or method of manufacture, could that be already covered by someone else's patent? So you may want to really consider that before you invest hundreds of thousands in a novel food submission, only to find out that the dose or the type of product you've got is already infringing someone else's intellectual property. So you might have just done a submission for them. So these are sort of key issues that are sort of fall slightly outside the actual this novel foods presentation, but definitely one of interest. And finally, use your right to free guidance. So what that means is you can contact the FSA, you can contact EFSA. You don't need a trade association. You don't need a consultancy like ourselves or a law firm to actually contact these bodies to give you guidance. You can reach out to them already. You already have that in your own rights. Anyone can have an audience in front of a competent authority. You can have a sit down, a discussion of these issues. But bear in mind that the rules are changing. So from March next year, the minutes from these type of meetings will have to be published. So it's going to mean a change to how sort of novel foods dossiers are potentially constructed, what's discussed with these competent authorities or authoritative bodies like EFSA. So now is sort of a good time to get a submission in and still keep a lot of those discussions confidential. But again, don't feel that that you can contact these agencies. That's your right to do that. And you'll find that they want only good things for you. They want only safe products to be placed on the market. And if they can help you do that, they will do. You just need to make sure you ask the right questions, and you need to build in enough time to get those responses. Okay, I hope that you found that useful. If you've got any questions or points for clarification, you can definitely sort of reach out to anyone at Legal Foods, including myself. The general contact information is on there, but I would point you to sort of legalfoods.com for any further information. Thank you and have a great day.